let us go in the next kind of methods, that is a molecular based method. We know that till 80s or till 77 or this particular time, we have a lot of flow cytometry studies already existing in the marine microbiology. However, after the advancement of the DNA sequencing technique by the Fred Sanger in 1977, a lot of moment has shifted uh, to study the genome, to sequence the microbes in order to understand them better. And also, when the Carey Mullis has developed the PCR and it was becoming a widespread in 90s, then a number of study has happened globally. One of the very first study to understand the microbial community across the in the marine environment was done by Stephen Giovanni, where he performed the first microbial community study by 16s rRNA library. I will explain you what the 16s rRNA library is in a couple of slides later. But since this particular time, maybe close to 90s, we have a lot of studies where we have been using the 16s rRNA or other molecular markers to study the sequence-based and function-based metagenomic studies. And then from 1998 onwards, we have a sudden increase in the in the molecular and the genomic techniques, thanks to the development of the next generation sequencing as well. Uh, a number of samples were getting sequenced frequently. For example, I was telling that ocean sampling day in 2014 and 2015, that has been sequenced across the world. That has given a lot of idea that about the diversity of the marine microbes. In addition, we also have oh, it will come in a later part of the slide maybe. So let us understand that how this molecular method works in order to understand the marine microbes. So you can see that all these microbes, microbial cell, do have DNA, right? And what you do is that you extract the DNA and then you look into the sequence. And based on the sequence, you identify them. Or based on the sequence, not only identify for the biodiversity, but also for the functional aspects. Okay. Steps includes, first is sample collection, that we have already seen that how do we collect the sample. Second is DNA extraction. So depending on the kind of the microbes you have been working, there are several DNA extraction protocol that you have to follow in order to get a high molecular weight DNA, right? Good quality DNA. After the DNA extraction, you have to amplify the target. So what I mean by amplifying a target is that you have to choose a molecular marker. So molecular marker, in case of, let's say, bacteria, is 16s rRNA gene, right? In case of other organism, it can vary. So basically, what we need to understand that you amplify that particular area using polymerase chain reaction, that is PCR. Okay. Once that is done, then you sequence it. Here I have given an example of Illumina based next generation sequencing, but you can sequence it with ABI Sanger sequence or maybe any other high throughput sequencing machines. Okay, then you do the bioinformatic analysis in order to get the result. Let us see in little detail. Okay, this slide I will come later. So what happens is that in the molecular method, one of the most important concept is metagenomics. So metagenomics, what it happens is that you do DNA sequencing, or you sequence the whole genome present in the water sample, okay, or present in the sediment or the environmental sample. So you take the sample, you do the DNA extraction, and then you sequence it. Once you do the sequence, then you will get to know that what all different kind of or microorganisms are present in that particular area. Two different approaches are taken in the metagenome. First is sequence based metagenome, and second is function based metagenome. In the sequence based metagenome, what we do is that we try to find, or we try to utilize the sequence what we are generating for the biodiversity study, mainly for the biodiversity study. Two different kind of metagenome can be think. First is a targeted metagenome where you use a molecular marker, for example 16S rRNA gene, or otherwise you completely sequence the DNA sample and then look into the what all different kind of organisms and genes are there. So what is the difference? Here you are only concentrating on the 16S rRNA gene. It means that most of the result what you will get is for bacteria and archaea. Whereas in shotgun metagenome, you are not separating any species. So you will get a gene not only for 16S rRNA or bacteria archaea, you will get a whole lot of whatever organism has been present there. So be it a eukaryote, be it whatsoever it is there, and whatever gene is present there. Okay, so that is called as shotgun metagenome. We will see the process in the next slide. For the function-based metagenomics, which we are not going to cover in this particular talk, what happens is that you use a particular functional gene. For example, yesterday I was giving an example for the archaea, that is ammonia monooxidase gene, ammonia oxidation, right, in the nitrogen cycle for archaea. So if you take that particular gene and then try to see that what all different kind of archaea is there doing the ammonia oxidation, then that can be classified as function-based metagenomics. In any of the methods of metagenome, as I said, that you have a sample collection, you have a DNA extraction, you have a sequencing approach, you have a bioinformatic analysis, and finally all the results are deposited in a public database, right? So let us look again in a more clear way this particular step, that is a PCR amplification of any molecular marker, or you can also do with the housekeeping gene. So molecular marker, it's a fragment of DNA that is associated with certain location within a genome. Okay, so suppose if this is a chloroplast DNA of any uh, plant, or if it is a mitochondrial DNA for any animal, or if it is a ribosomal DNA for any fungi. Okay, so any region which is available here, for example here 18 is rRNA. Okay, and if this you are going to use for the for reducing the microbial community, this can be called as a molecular marker for that particular group. Okay, so it is a part of a genome, or if it is located at a certain location within the genome. Okay, and this you will use if you are using nuclear DNA mostly for the bacteria. Let's say, let's not go much into other organisms, let's stick with the bacteria. Then you will look for the 16S rRNA, right? Now what the 16S rRNA means is, okay, another thing is that if you have chosen a molecular marker, let us suppose you have chosen 18S, or if you have chosen 16S, or if you have chosen, let's say, uh, RPO, B, or any reason, maybe D loop, okay, any reason. So that area which you are amplifying in PCR, it's called as amplicon, okay, and the sequence, when you do this particular sequence, it is called as amplicon sequencing. Okay, so amplicon is nothing but a DNA or the RNA piece which you are going to amplify in the PCR. Okay, in case of bacteria, we choose 16S rRNA gene, which is very much universal for to understand the bacterioplanktonic community. Okay, so 16S rRNA gene is associated with the small subunit of the ribosome. Okay, and we choose this particular gene because there are several conserved sites and then several very hypervariable regions. So in the conserved site will be used for the designing of your primer for the PCR, and for the hypervariable region will tell you uh, the changes among the different species of the bacteria, because that is what you require, right? If you want to know that what all different species are present in the environment, you need a region which is having a conserved site for the synthesis of the primer, and at the same time it is having a region which tells you the species, right? So this 16S rRNA, it's a very good site because it's a combination of conserved variable and hypervariable region that evolve with different rates and enables the resolution of both very ancient lineages, right? So this is one of the major reasons that why we are using the 16S rRNA gene. Further look into how the 16S rRNA genes are located. You have a V1 region, that is a variable region. So this variable region is evolved in the different species at a different rate. So you will have a lot of variable region here, but in between you also have a conserved site. So these conserved sites will be useful in order to make the primers, because you remember in the DNA replication what it happens. A primer will go, or a small sort of a small nucleotide stretch will go and attach, and then it will initiate the uh, replication, right? Similarly, in PCR, you have a primer which will go and bind to the DNA, and then from there your replication starts in both the direction, right? So it is this particular small segment of oligotype which is required to be made in this conserved region. Okay, so overall it's a 1495 base pair, 1465 base pair gene. So 1465. Okay, and in this 14, so 1465 means, let us suppose if it is a T, okay, G, C, only four bases are there, right? So A T T G C. Likewise, there are 1465 bases. In between, some regions are very much variable, which helps in identifying the species. So there are 9 regions in 16S, and at the same time, you have a conserved region where you make the primers for the PCR. Okay, that is why the 16S rRNA gene is very much used for the bacterioplankton uh, community analysis, right? See, one of the work from our lab we have done recently from the salt pan, and when you use the 16S rRNA, you can find. So what happens is that you did the PCR, right? After the PCR, you did the sequencing, as I said in this particular slide. Wait, in this particular slide you did the sequencing. So in our case we sequenced it with the Sanger sequence, old method, and then we did the bioinformatic analysis. So when you sequence it, you will get a DNA sequence, right? ATGC, CGTA, whatever it is, right? And that you use the bioinformatic analysis in order to find out that what all different organisms are present here. So in here you can see that we have got one Staphylococcus species, we have got two Halobibrio species, and then we have got close to 6 Chromohalobacter species, and all are sharing some common ancestor in past, right? And then finally you have archaea taken as an outgroup. Why we take this? Because we know that evolution has happened in the past. So so considering that all the organism are originating from one particular species, right? In some certain point of time one species is separated, and that has formed a different domain called as archaea, right? So you know that the evolution, it gives the directionality. So now you know that evolution has separated the bacteria from archaea. Now within bacteria, you do the bioinformatic analysis to find the relation between these organisms, uh, how they are, and that can be done with the 16S rRNA. So using the same gene, you can find out that different kind of bacteria belongs to different uh, genus or species, okay, using the phylogenetic analysis. Now the second method is shotgun metagenome. So as I was saying that in shotgun metagenome, what we do is that we collect the sample, so maybe a water column or a sample or sediment sample, you extract the DNA, and then you do the full sequencing in one time, okay, without without doing the rRNA gene PCR or any other PCR. So we skip that step, and then we did the DNA sequencing directly. Once you do the DNA sequencing directly, you will have a tedious bioinformatics process, because now that you have gene from many different organizations, you have genes of different classes, different functions. All together, you have to do the bioinformatic analysis, after which you can find out different genes. So you can find out different microbial species. You can also find out different pathways, I mean genes involved in the different pathways, right? And that helps you to find out uh, biomarker discovery, microbial based prediction tools, and so on, phylogenetic constructions, and so on and so on, right? So that is the process of shotgun metagenomics. One such example, a very early article, even yesterday I shared, that using the environmental genome shotgun sequencing, metagenome, in Sargasso Sea, which was the very first paper by Craig Venter et al., okay, has found that close to 1800 unique genome was present in the marine water, belonging to 48 unknown bacterial phylotypes and 1.2 million previously unknown genes. So it means when you do the metagenomics, there is a ample chances that you will get previously unknown set of microbes, unknown set of genes, unknown functionality of the genes, and it helps to discover a lot of new information. For example, it is this metagenome which helped to find out the SAR11 bacteria, which yesterday I was telling you is a very small size bacteria, ubiquitously present across the world ocean. We were not even knowing that that bacteria is abundantly present. It was very difficult to grow that bacteria in the culture, again because it is present in the oligotrophic conditions, right? So after the metagenome revealed that there is a certain group of bacteria which is present, okay, uh, people have started using the idea that from where this bacteria is isolated and trying to give the nutrient condition based on the sample collection location. Okay, so now suppose if I know that I have collected the bacteria, let's say from Marina Beach, and then in Marina Beach I know that what could be the possible chemical composition of the water in that particular environment using different analytical techniques, right? I will try to create the artificial medium according to that, and then try to grow the bacteria. If the bacteria gets what they really use in the natural environment, they will grow. And this metagenome has been successfully utilized for several species, I mean, to grow in the laboratory conditions, which were previously difficult to grow in any of the known media. Okay, so this sort of benefits are there for the metagenome studies, right? And we already know that the result from our group I have shown you was used by this particular method, that is a capillary sequencer by Applied Biosystems, developed by the Fred Singer, right? Automated sequencing. However, in the last 15 years, we have a great revolution in the sequencing technologies. We not only have a second generation sequencer, we also have third generation sequencer. So in second generation sequencer, these are called as, these are very high throughput sequencing, but produces a small fragment of the genes. So it is very difficult bioinformatically to annotate them and to make a meaning out of those sequences. However, it is being, that Illumina sequencing is being used in a great deal across the world. Okay, slowly we are moving to the third generation where we have the PacBio. So PacBio is again producing a lot of genes at the same time, sequencing the whole genome at the same time, and producing a bigger fragment of the genes. So it is a little bit difficult. I mean, a little bit easy uh, to actually sequence the whole genome. However, it is a very costly approach. This particular PacBio. There is another one which we are also using, that is Oxford nanopore sequencing. It is a very cheap method because the whole machine set will come in close to 10 to 15 lakhs, and it also produces a bigger fragment of the genes. What I mean by bigger fragment, when you use the second generation sequencing, you will get 100 to 200 sequences in one stage. And then using bioinformatics, you have to find or make a proper big gene piece by aligning this 100 to 150 in a very cumbersome method, right? Whereas in this particular method, you will get more than 1 kb or sometime even 10 kb or 20 kb length, right? So in one go itself, you can have the whole gene sequence, right? So this is what I was saying, that at present we have been mostly using the Illumina sequencing, and slowly people have started moving to the third generation sequencing, which produces the long read, right? As a result, you would see that till 1996, we have maybe first or second, I mean, one or two bacterial or microbial genome sequence. And at that time, those sequences, if you do a genome sequence, it would appear in a journal like Nature or Science or Cell, right? However, down the line, 20-25 years, we have more than 3 lakh microbial genome in NCBI database. So I hope that you are aware that NCBI database is a public repository for not only for the genome but all sort of molecular data which you generate in the form of genes, in the forms of whatever, like genome, transcriptome, uh, uh, any study you do, you have to keep it in a public repository, and that you can see that close to 3 lakh or 3 lakh 10,000 close to genome for prokaryotes have been already there. And that also you can differentiate with the virus, with the plasmids, with the organelles, and for the bacteria. Okay, so I hope that now I have covered what could be the sampling methods, what are the various methods for cultivation of bacteria or the microbes, imaging techniques, what are the various molecular based methods, not directly the 16s but also used in the flow cytometry and used in the fluorescent in situ hybridization. Okay, that is fish. And then we have also seen genomic techniques, evolution of the genomic techniques, which has made life easy to do the metagenomic studies. Okay.